Welcome, thanks for coming today to the defense of Julian Conley. Julian comes to us from Texas, so he went to the University of North Texas, has a keen interest in the environment and particularly aquatic environments. And so came here wanting to study turtles. That was something we talked about. And then the pandemic happened, and a week before he got to go to the Smithsonian, we found ourselves having to figure out, well, what are you going to do now? And amazingly, we've come up with a really great project here, and I think you're going to see something that I think Julian's done a really fabulous job, and he's going to tell you all about the community structure for turtles at the Great Fossil Site. Most of you have been to these before, but the structure for these is that Julian's going to present for about a half hour. After that, the committee and the audience will ask questions. Then we'll ask everybody to leave, and we'll maybe ask him a few more questions, and then have him go out as well. So please reserve any questions till the end of the presentation. I'll leave it to you, Julian. We're recording, correct?
Yes.
Okay.
All right. I'm gonna step off of this camera changes. That's cool. All right, so hello everyone. My name is Julian, and I'm presenting my thesis today on community structure analysis of turtles with application to the Gray Fossil Site, which you may have heard of. Yeah, so turtles are an ancient group of reptiles with origins in the mid-Triassic, and they had their first major diversification events in the mid-Cretaceous. That's when a lot of the families that we know today came about through to the Eocene, a lot of diversification. Now, today there are over 350 species of turtles, and they keep splitting them. There's more diversity now, but they consist of two major suborders. Those are the Pleurodires, or the side neck turtles, and the Cryptodires, or the hidden neck turtles. There are 14 total families of species, 5 of which currently live in the United States. Now, the most recognizable feature of turtles that everyone's familiar with, of course, is their shell, composed of a carapace and a plastron fused together. The shell has been really a key feature in their success over the years. It's also limited them ecologically to what they can and can't do. However, they have diversified into a broad range of habitats and niches. Most turtles are freshwater aquatic, to some extent. There are terrestrial turtles as well as marine turtles. It's not uncommon to find many different turtle species living together in the same environment. This is because they're pretty successful at adapting to different niches, different life strategies. So you can find communities of them in large numbers of different species. And turtles are pretty important for the environment. Aquatic turtles can quite often make up the highest proportion of animal biomass in an aquatic ecosystem or habitat. So they're major controllers of other aquatic organisms. And then of course we have things like gopher tortoises, which are ecosystem engineers creating habitat for other animals in those environments. So turtles are really important key features of their ecosystems. So the context of my study was the Gray Fossil Site, which of course is right here in East Tennessee. It's an early Pliocene deposit, so about 4.5 to 5 million years old. And it preserves an ancient pond, a sinkhole that infilled. Now the evidence we have of the plants and animals around that pond suggests that it was a forested environment. And those, along with the isotopic evidence, suggest that it was a much warmer climate than today. There's a mid-Pliocene warm event which occurred after the Gray Fossil Site, but it was already trending upwards in terms of temperature. So you have a warm, humid, subtropical climate in a forested aquatic ecosystem, which is just about the best thing turtles could ask for. So there was a diverse ecosystem or community of turtles at the Gray Fossil Site. We know of at least 8 species for sure, possibly more. And we did have all 5 of those modern, all 5 of the families that are currently living in the US did have representation at the Gray Fossil Site. So we see some of the species that we would expect to find here in East Tennessee today, things like Calydra, the snapping turtle, some pond turtles like Chrysomes and Trachomes, the painted turtle, slider turtle, box turtles, of course. And we also find some species that we don't expect to find here based on current distributions. Things like tortoises, gopher tortoises, those today live further to the south. And then things like Emidoidea, the Blandings turtle, today are restricted to the upper Midwest. So they don't have any range overlap currently, but they did overlap at the Gray Fossil Site. Now, just within the last year, we found soft shell turtle fossils, Trionychid. So that completed our data set for all 5 modern turtle families, which is great. So fossil sites like the Gray Fossil Site offer us a chance to look not just at individual taxa organisms, but entire ecosystems that existed in the past. One method of doing that is called community structure analysis. And that's essentially looking at the ecological composition of different niches, proportions, and abundance of the niches that all the different animals and organisms in that community are occupying. So you can do this with taxonomic composition, with niche composition, lifestyles, things like body size, diet. And typically when this has been applied to the fossil record, it's been done with mammals. Works really well with mammals because mammals have really good signals for their ecological relationships in their morphology. So things like dentition are signs of diet, locomotion. You can see the limb elements, how they're moving through the environment. So it works really well with mammals. It's been applied to Mesozoic mammals, as well as Pliocene African mammals in a hominid context. But it's never been done with turtles, at least with turtle fossils before. So my project was to take those methods that have been applied to fossil mammals and apply them to fossil turtles at the Gray site. So what I really wanted to show is that because of the climate of Gray was so much warmer than we expect today, more of a subtropical sort of Gulf Coast type environment, that the turtles at the Gray fossil site, the community structure, should resemble that of modern southern subtropical Gulf coastal environments. I also wanted to demonstrate that turtles are good indicators of past environments. Typically, when turtle fossils are found, they're used to say, okay, yeah, it was warm and maybe aquatic here. And that's about the extent to how much they get used. And then they get tossed away because paleontologists often don't want to find turtles. They find them not very exciting. So I'm here to change that. We also wanted to identify some trends in the biogeography of modern turtles. Can we compare different regions, different areas and habitats and see different trends in the ecology and taxonomy of the turtles that are occupying those spaces? So why is this important? Why are we concerned with turtles and their ecosystems? Well, unfortunately, turtles are not doing very well. The future is pretty bleak for turtles. Currently, they're among, if not the most endangered groups of vertebrates on the planet. And if you look at this graph, which came from a recent study, you can see that across the board, throughout most habitats, in fact, yeah, all but one habitat, turtles are by far the most endangered group of reptiles. And this pattern holds true if you compare them to other vertebrates as well. Now, some of the major threats for turtles are habitat loss, of course, humans coming in and doing whatever we do. Things like invasive species, so Trachomys scripta, the red-eared slider, constantly getting introduced into all these different environments, pushing out native turtles. And then, of course, the big one is climate change. So, turtles like warm weather. So climate change should be good for them. But what they don't like is the weather changing or the climate changing very quickly, environments being altered much more rapidly than they can adjust, than they can adapt to that. Especially turtles with smaller ranges, things like bog turtles, very restricted home environments. They're going to have a lot of hard time adapting to those changes in environments, especially with the other pressures being put on them. So to do a community structure analysis of turtles, we have to have the ecological variables to apply to them, to put them into these categories, discrete classification systems. This is challenging with turtles because they're not like mammals and they don't really want to do one specific thing. They're very generalist, very adaptable. So it's hard to say for sure what the difference is in the ecologies of different species of turtles. So essentially what we have to do, or what I had to do, was look at a spectrum of different turtle behaviors and the nuances of their microhabitats, feeding ecologies, and figure out where on those spectrums can I divide these to give both specific enough classifications that they're meaningful to the data, but also broad enough that I'm not creating an artificial bias by splitting them up too much. So the classification system I came up with looked at 6 major ecologies or niches that they can occupy. Then I also used taxonomy. But for the ecologies, of course, there's body size. So there's 4 even body sizes, the length of their shell, essentially. Habitat, so aquatic turtles or semi-aquatic turtles are those that are primarily using aquatic environments to get food resources and using that space. Terrestrial turtles, things like tortoises, of course, spending most, if not all of their time on land, using terrestrial resources for food acquisition. And then semi-terrestrial turtles are kind of in the middle. They're going between the two, maybe riparian zones, bogs, things like that. For the semi-terrestrial and aquatic turtles that are using those aquatic resources, I wanted to know more about their microhabitat within the aquatic environments. So things like lentic, lotic, Pretty easy distinction there, except there's a lot of generalists that use both. So we have lentic, lotic, and generalist, and then substrate. So what is that benthic layer? What kind of benthic area do they prefer? Some prefer fine grained, some prefer coarse grained, or pebbles, bedrock, things like that. So getting into the details of the microhabitats of aquatic structure. And then for the semi-terrestrial and terrestrial turtles that are living on land using land resources, I wanted to know, are they using more closed environments, woodlands, forests, or open environments, plains, prairies, deserts, things like that? And then for the diet, that one is a little tricky because all turtles, every single turtle is an omnivore, but some are more omnivorous than others. So when I say herbivorous or carnivorous, what I really mean there is that their feeding ecologies reflect specific adaptations. Tortoises, for instance, will eat meat, they'll eat bones and things, but they're primarily adapted for eating plants. Whereas an alligator snapping turtle is happy to eat an apple if you throw it in there, but they're primarily adapted for prey acquisition. So they would be carnivorous. Now, if we look at this handsome little guy down here, this is a spotted turtle. This is one of my favorite turtles of all. Just look at him. He's so cool. He has a body size score of 1, so it's a small turtle with among the smallest turtles in North America. It is a habitat of 2, so semi-terrestrial. So this is going between aquatic and terrestrial habitats. Now, for its aquatic habitat, it does prefer lentic and fine grained, so think small, soft ponds without any current. And they're terrestrial 1, so located in woodlands, forests. And then finally, a diet score of 3. So this is an omnivorous animal. It's not particularly picky about what it eats. So I did that for every single turtle species in the United States. Then I had to apply it to the gray fossil site turtles. This is where it gets a little tricky because unfortunately, all the turtles at gray are dead. So we can't observe their living behavior. We can't watch them in the wild and understand their ecologies directly. So we have to make inferences based on their fossil morphology, as well as use proxies like modern turtles that are close relatives of those extinct turtles. This is pretty straightforward with things like trachemes that are morphologically consistent with the modern trachemes. So for the most part, turtles are pretty conservative ecologically within a genus. They also have slow rates of adaptation or evolution. So we can be reasonably confident that these turtles are similar enough to the modern turtles that we can use those turtles as a proxy for the extinct turtles. And also my ecological classifications had enough leeway that I felt there wasn't going to be, a little bit of variation is going to make or break the data too bad. But we will talk at the end about some of the exceptions to that, some caveats. Now that I have my ecological classifications, I needed some community data to reflect actual communities and ecological compositions of modern turtles. So I went to the National Park Service website, went to their database and looked at species occurrence per site for every site in the United States and pulled every single one that had 2 or more turtles, so like a community of turtles. And then looked at, so each site has its turtles and its ecological composition of turtles, and then proportions of the ecological of the niches at that site. And I did have to correct the data a little bit. I had to update some taxa names that were outdated, as well as exclude things like trachemes that were occurring way outside of their native range. And what I ended up with was 130 national park sites. And then I added in like 3 or 4, 3 national wildlife refuge sites using iNaturalist data, just to get a little bit of better coverage of some of those specific regions. Now, I didn't want to compare every single site to the gray fossil site individually. So I grouped those modern sites into 3 distinct grouping types. We had level 1 ecoregions, level 2 ecoregions. Ecoregions are EPA designated sort of biomes, areas of similar biome, climate, and geology. Level 2 nests within level 1, so they're just more specific. And then climate regions, and these are broad categories of temperate, cold, arid, and subtropical that just reflect things like temperature and precipitation. I also collected data for every site, the latitude, maximum temperature, minimum temperature, and annual precipitation to get an idea of what that specific community is, what the conditions there are. So for the statistical analyses, I did a descriptive statistics, which is essentially just visualizing the data, the proportions and abundance of different niches between ecoregions using bar graphs, which we'll see in a minute. I also ran a discriminant function analysis test. So this is essentially grouping, taking all the groups, all the ecoregions or whatever, and looking at the composition of all the different sites within those and those ecological compositions, and trying to compare them to other groups, trying to find the best way, the best discriminant functions that can tell the different groups apart. It can just discriminate the different groups. And then it takes an ungrouped site, which in this case was the gray fossil site, and predicts where gray would land, which of these modern groups it best matches with. And then I ran a bivariate correlation analysis, which looks at the ecological variables of the turtles and correlates them with environmental parameters to see where there are significant correlations for the turtle ecologies. So for the results, for taxonomic composition, we have gray fossil site over here. I remember gray is one of the places where we find all 5 modern families. And now it's difficult to find clear matches. But if we look at this one, 8.5 is the Mississippi alluvial Gulf Coast. It's kind of similar. It does have all 5 present. However, has a much lower proportion of turtles, higher proportion of Amidids than what we see at gray. If we look at 9.5, that's the Texas, Louisiana Gulf Coastal Plains. This one doesn't have callidrids, but it does have a much more even balance of the other 4, especially tortoises, much closer. But what we do see is that there aren't really any specific clear matches with taxonomy. If you look at habitat preference, obviously most turtles are aquatic. The gray fossil site, based on our preliminary interpretation of some of the turtles here, does have a much higher proportion of terrestrial species, which stands out with some of the southwestern sites, things like the warm deserts and Sierra Madre or something, that also have a lot of terrestrial species or proportionally. What I find interesting is that the semi-terrestrial species are all restricted to the east, and they're especially abundant in the northeast, as opposed to the terrestrial turtles. Now for aquatic preference, lentic versus lotic, again, most are generalist. I do see some trends here with the gray fossil site has a higher lentic proportion than most, and that's going to match it up with, again, the 8.5, the Mississippi alluvial, and the Texas, Louisiana Gulf Coastal Plains, as well as 15 over here is South Florida, so the Everglades. So we're getting coverage of the entire Gulf Coast, has similar trends in lentic species. And also lotic species are really prevalent in the west, southwest, and Great Plains areas, which I found interesting. For terrestrial preference, this distinction is really clear. It's almost perfect. The east has closed species, the west has open species. I don't think I need to explain why that is. Now the gray fossil site, it does kind of match 9.2. This is the northern Great Plains where box turtle species overlap. However, this is the one graph where I would take this with a grain of salt. Our understanding of the biology of the tortoises at gray is still preliminary. So this is based on the modern proxies of the tortoises, where some of the freshwater turtles we have a much better understanding. So I'd be a little wary of this one. Now for body size, this one's very good. We see a clear match between gray fossil site and the 9.5, the Texas, Louisiana Gulf Coastal Plains. They're very similar, as well as the Mississippi alluvial, and again, that South Florida Everglades. So again, the body size and proportions are matching up really well with the southern Gulf coastal ecoregions. And then for diet, the first thing that stands out is that gray has a huge proportion of herbivores. Again, tortoises are herbivorous. So this kind of matches it up actually with the warm deserts over here. However, the warm desert also has some mesoherbivores and a huge proportion of mesocarnivores compared to gray. So probably a better match for gray would be 9.5. The Texas, Louisiana Gulf Coastal Plains, again, is really the most similar to the gray fossil site. And it's worth pointing out this Mississippi alluvial and to a lesser extent, the Everglades also have similar trends in the dietary preferences to the gray fossil site. Now, if you look at the climate regimes and we look at the niche abundance and the different sort of ecological compositions of the different climate regimes across some of our variables here, we can see that gray fossil site really closely matches the subtropical regions. And it's consistently showing the same shape, and it's, you can see it, especially with the lentics over here and trending towards the mesocarnivores. But it's very different, very distinct from the temperate, cold, and arid across the board. I picked these 4 variables just because they show it the best, but we see the same pattern in the other 3 as well. So for the discriminant function analysis, I got statistically significant results for all the different ecoregions and climate regimes. So the test worked, which is good. The low Wilks lambda scores mean that there's not a lot of overlap between the two, especially with the level 2 ecoregions where it's splitting them up so finely. Now, correct classification is telling us how well does it predict each site to the correct group based on the model it comes up with. Level 1 in climate regimes get 80%, which is excellent. Level 2 gets 50%, which is less excellent. But if we look in the actual data results, this is kind of biased by some of the southwestern sites with lower sampling. They're doing very poorly, whereas the eastern sites with more sampling, they're doing really well. So it just depends where we're looking at on the map, on the data. The gray fossil site, when it got plugged in and it got predicted to each of the groups, for level 1 ecoregions, it got predicted to the Great Plains, which isn't really what we would expect based on what we understand about the gray fossil site environment. However, the test gave it a high posterior probability, so it says, yeah, it looks most like the Great Plains, but a low conditional, meaning it's still not really fitting with them. It's just closest to them compared to everything else. When it did the level 2 ecoregions, it predicted it as the Texas, Louisiana Gulf Coastal Plains. Remember, that's the one that in our descriptive statistics kept matching up with. And it gave it a high probability and a fair conditional. So yeah, it probably is pretty close or within that Texas, Louisiana Gulf Coastal range composition. And then climate regimes, unsurprisingly, subtropical, absolutely. The conditional was like 99.8%. So it was very confident that the gray fossil site was landing not just near, but within the range of subtropical sites. Put that on a map so we can visualize that. We have the gray fossil site up there in Tennessee. For level 1 ecoregions, the Great Plains. So anywhere with a 9 on it, essentially that's from Canada all the way down to the Gulf of Mexico. That's not particularly, doesn't tell us very much about the ecosystem at gray. The climate regimes gave us a subtropical result. So that's like southern Florida Gulf Coast region where it's warm all year round, not really freezing temperatures at any point during the year. And notice that there's an overlap spot right there in South Texas. And that's where the level 2 ecoregions predicted the gray fossil site would best match in that 9.5, the Texas, Louisiana Gulf Coastal Plains, all the way from Mexico by the Rio Grande up into Louisiana by the Mississippi Delta. So what we're getting from that is that the gray fossil site does resemble subtropical sites along the Gulf Coast, much further south than today. Now, if we look at what the discriminant functions gave me, over here, these sites are most of the sort of arid southwestern ones. Up here, we have some of the Great Plains semi-arid regions. And then down here are most of the sort of higher precipitation, higher latitude eastern sites. And gray is sitting sort of right in between the Great Plains and the eastern higher latitude or higher precipitation sites. So it's not quite fitting exactly with either one. You can see some of those Texas, Louisiana sites are actually pretty well spread out. And the closest site to the gray fossil site is one of those Mississippi alluvial sites right here. So again, probably right around here. So for the bivariate correlation analysis, there were some statistically significant correlations, especially between the taxonomic, the family groups. So tortoises, mud and musk turtles, and testudinids and chinosturnids, they prefer low latitude, warm temperatures, especially tortoises. They just don't go above a certain latitude. They don't go past a certain minimum temperature. Things like callidrids, snapping turtles, they proportionally had a correlation with higher latitude, cooler sites. This is not because they have higher diversity up there. It's because there's lower diversity of everything else. They're just more tolerant of those environments. Larger species did tend to trend to lower latitudes despite the snapping turtle. And this is due mainly because, remember, turtles are ectotherms, so they rely on external temperatures for all their metabolism and biological functions. So they rely on warmer climates. The omnivores, which primarily driven by amidids, they preferred higher precipitation, whereas in the more arid western areas, you can see more mesocarnivores like mud turtles, softshell turtles, and then tortoises, so herbivores over in the west. And I found it interesting that softshell turtles, like this guy here, they didn't have any strong correlations with environmental parameters, like temperature and precipitation. My interpretation of that is that they're likely more controlled by the presence of lotic environments. They really like rivers. So that's probably really what's driving them. For the extant turtle communities, we do see that there are strong trends between different regions, biogeographic patterns with their ecologies related to the environment, especially within those ecoregions. So a lot of lentic turtles prefer the east, lotic species prefer the west, at least proportionally. This is likely because ponds and things are not stable year-round in really arid western areas, but they're going to be more reliable environments in the east for this high precipitation. Whereas those western taxa are going to be relying more on rivers, river systems for their habitat. Terrestriality trends, very low latitudes of tortoises. They don't like cold, so they just don't get up here. And then semi-terrestrial turtles, these are mostly amidines, things like bog turtles, spotted turtles, wood turtles. Those do have a higher latitude trend, as well as a more eastern higher precipitation trend. So closed environments, things like that. Some turtles are just more tolerant of environmental variability than others. We don't see things like tortoises, spreading all around. We do see painted turtles, snapping turtles, box turtles. Those are really common everywhere. They just have really good tolerance. Whereas some other turtles, especially this here, Malaclemes, diamondback terrapin, is going to be really specifically, restricted to certain habitats. In this case, estuary and coastal marshes. So you're not going to see it spreading throughout the continent. Now, what does this say about the Gray fossil site? Well, community structure analysis of turtles at Gray was successful. We were able to show that, the turtles at Gray did have a distinct ecological composition different from the turtles that exist here today. And they're more similar to those southern Gulf coastal areas. So the Texas, Louisiana Gulf coastal plains has the most similar ecological composition of turtles to the Gray fossil site, what the Gray fossil site had. However, taxonomically, the Gray fossil site is really unlike anything that we see today. Not even the Texas, Louisiana, that area, it doesn't have the same structure of families and species that we see at Gray. So especially the tortoises and the Emidoidea that I mentioned earlier, those are species that don't have any range overlap. They're like 900 miles apart today. So if we looked at Emidoidea's modern distribution, we would conclude that it's probably preferred cooler climates around the Great Lakes. However, in the past, clearly that was not the case. It did prefer warmer environments. That suggests that maybe things like the Pleistocene glaciations have impacted the ecologies and the distributions of some of these turtles. Now, this guy here, you can mention him. This is the Coahuilan box turtle, which is a little bit different from the box turtles we have here in the United States. It's more aquatic, whereas ours are more terrestrial. And there is some indication morphologically that the Gray fossil site turtles may have been similar to the Coahuilan box turtle. And we just need more research done on that to be sure. But don't worry, I did rerun the analysis, changing the Gray box turtle to be more aquatic, and I got the same results. So I wouldn't worry too much about that affecting all the results. Now again, turtles are highly endangered pretty much everywhere. So what does this have to do with the Gray fossil site? They're all dead anyway, it can't affect them. Well, because we don't really understand how things like climate change are going to impact turtle communities in the future and how they're able to use different environments, having a sort of window into the past, into a different ecosystem at a different time period, under a different climate regime, especially one that's warmer than today, gives us maybe some indication of how modern turtle communities may respond to these environmental changes in the future, which gives us a better ability to prepare for that, especially for turtles, again, like the bog turtle, that have very restricted environments. We want to know where their habitat's going to live, where should they live in the future, where are they capable of living, and looking at past fossil ecosystems gives us a better perspective on that. So for future research, I'd really like to apply this to other fossil sites, especially contemporary ones with Gray, things like Pike Creek, Indiana, Monbrook and Love Bone Bed in Florida. I think it'd be interesting to get a broader geographic spread of sites to see if we can replicate this in a meaningful way. I also want to do it again with different regions, different geographic groupings. Here are freshwater ecoregions and turtle bioregions that this could apply to really in a really interesting way. I also want to expand the data set, refine the data set. I think the more I learn about turtles, the more I come to understand their lifestyles and their ecologies and the nuances, really what turtles are and how they live. So I think that between the time I started this project and now, I could probably come up with some more interesting ecologies that reflect really the nuances in microhabitat use to get a better understanding and a better perspective on turtles across the environment as well as expanding this data set beyond the United States to encompass global turtles. I think that'd be really interesting. It's not just fossil sites here, there's fossil sites all over the world that this could be applied to and could be really, really meaningful and helpful for turtles in the future. So I'd like to acknowledge my advisor, Josh Samuels. So he was a huge help when my first thesis project collapsed, crashed and burned during the pandemic, or right at the beginning of the pandemic. He helped me get back on track, helped me find a new project that I really enjoyed, really liked this project. So thank you, Josh. I want to thank my committee members, Chris Widga and Blaine Schubert, for being very patient, as well as helpful, giving me some advice and input, and just really having confidence, having faith in me that I would get this done, during that dark 2-year period. I also want to thank, of course, the staff at the Gray Fossil Site. You guys make these fossils available for us, whether you're getting them out of the ground, preparing them, keeping them in collections. We couldn't do this without you. I want to thank Andrew Joyner for helping me with GIS when I was getting all the sites, mapped out and finding their ecological or environmental parameters. I was completely stumped to spend hours trying to figure it out. He came in and there it is. Stephen Jasinski, of course, he's my predecessor in turtles at the Gray Fossil Site. He provided some advice, some input on the specific ecologies of the Gray Fossil Site turtles that really helped this project. Of course, all my fellow grad students, whether I started with you or whether you started before me or whether you started since then, you guys have made this a much better experience than I could have hoped for ever. So thank you guys so much. I also want to thank all my friends and family at home for putting up with me for 27 years while I talked about nothing but dinosaurs and reptiles. So thank you guys. And any questions?
So you made a mention of including a larger data set, including animal or environmental data from other parts of the world as well. And one of the things I was wondering is, do you think particularly in Southeast Asia and China, that would be a pretty important comparison to make, considering how many overlapping species we have with them, even in the present, but then also in the past with cryptobranchs and alurids.
Oh, that was a really good question. I actually had not thought of that, but you're absolutely right. So based on the mammals and the plants that we have at Gray, we know that there's a lot of similarities to Asian sites, especially temperate Chinese areas. I think, Sarah, you found some comparisons with the mammal community. That absolutely could be the case. Asia has a huge population, especially of geoamidids, which we don't have in the United States. There is one species of geoamidid, or a genus of geoamidid in Mexico, which is why I had to say United States, not North America, because there is a 6th family in North America, just not here. But there's a lot of geoamidids in Asia, which are comparable to amidids. There's also a lot of soft-shell turtles. So we could see similar trends. There is even an Asian box turtle. It's not the same genus. It's not related to our box turtle at all. But we could be seeing similar ecological trends. That is a really good point. Also, there's something called the Asian turtle crisis. Most of the most endangered turtle species in the world are all from Asia. It's looking pretty grim there for them. So that would be a great place to apply this and try to get a better perspective on the community structures there. What's going on with this?
Thank you. Since you mentioned how this could be useful for understanding how turtles conservation will change in the future, have you thought about maybe projecting into different time periods using species distribution modeling and maybe combining that with a community structure analysis?
I have thought about species distribution modeling, especially with invasive turtles. So Trachomys scripta, red-eared slider, which anytime that you see turtles being sold as pets in the mall, it's a red-eared slider, guarantee it. So they're a huge problem. So doing species distribution models of those guys in places like Asia and Europe would be really important. You asking about different time periods?
Yeah, either future or the past.
We could do that on a sort of broad scale, seeing, on an ecoregion scale. I don't know about a super fine scale. So you need a pretty good understanding of the climate and environment of those time periods. I hope that answers your question.
Yeah.
So you mentioned kind of at the beginning of your presentation that a lot of our modern turtle lineages kind of appear in the middle of the Cretaceous period. Do you think the community structure analysis you did is like inherently kind of tied to the later Cenozoic, or could you use this as a way to model turtle ecology in really deep time, perhaps even into like the like Eocene or Cretaceous or something?
That's a good question. When I say mid-Cretaceous, it's sort of a trend that begins in the mid-Cretaceous. That's when soft shell turtles appeared. And I guess side necks that appeared before that. We don't see things like tortoises and amidids until the Eocene. So there's a huge window. All of those like chalydrids and kinosternids are evolved into in the late Cretaceous. What would make me really wary about trying this in like a late Cretaceous community is that there were also a bunch of other families that are now extinct, that have been extinct for 70 million years. So we would have to make some pretty big assumptions about their ecologies. Again, because mammals have the dentition, which indicates their diet really well. But for a turtle diet, it's much more vague. And so I would be wary about applying like dietary preferences to a turtle that's been extinct for 70 million years, which is a shame because it would be really interesting. But for now, I would stick to the late Cenozoic when we're seeing, maybe Miocene would be the latest or the earliest I go back to, when we're seeing some modern genera appearing. Cool. You had a question too?
What would you think about the potential of expanding your environmental categories to include other groups of organisms that are also present in the ecosystem? So like, for example, just thinking about Greg, does the presence or absence of crocodilians influence your turtle community? Is that something that you think you might be able to do with this kind of an expansion of this kind of analysis?
I don't see why not. If it's just a simple binary between yes or no, there are crocodilians here. That was something that Josh and I had talked about, is whether or not things like alligators are limiting factors on the distributions of turtles. Potentially they are. Haven't looked into that as much. But yeah, co-occurrence of other taxa, maybe even like prey species or plant species. It could be really interesting. Especially, I do think there's a strong correlation between diversity of turtles and alligators, just because alligators are so tied to really warm climates, low latitude, subtropical, aquatic systems, places that naturally are going to trend towards turtle diversity. So maybe not necessarily seeing them be a limit on the turtles or like a specific relationship, but there's probably going to be correlation here, I think.
So with this, we're starting to get a better picture of like what the environment at Gray would be like, what the ecosystem of the turtles would be like. But obviously Gray is a site that's actively being excavated. And obviously we haven't found everything that's here yet. Is there anything to indicate that all the data that you found or the connections between Gray and some of these other regions that suggest maybe a gap that in our turtle diversity?
That's a good question. So I talked to Josh and Blaine about this recently. I feel confident that this is probably reflecting a good portion of the turtle diversity, mainly because, we are seeing a pretty good spread. If we were seeing something crazy, like it's all kind of sternids, I would think, okay, there's probably something wrong here. But because what we're seeing is proportionally, I did say it was taxonomically non-analogous. That was more of the genera and species. Family-wise, it's not too bad. It's pretty much within line what we'd expect. We might expect some more amidids, but not really, I wouldn't need it. What's really sort of the biggest question about the taxonomy of turtles at Gray is that we have identified 2 tortoises. I didn't do it, someone did pass, and said that there are 2 tortoises. Now, this is not a tortoise morphology paper or study, so I sort of had to trust them, put my faith in that. I did rerun the model again with just 1 tortoise, and it turned out the same, just like with the box turtle. But I don't know about any major gaps. I mean, the Trionicid was a major gap that we filled. I can't imagine what it would have been like to do this study before that was found. Like if it was found, yesterday. then like, well, I guess I'll just drop out. I'm not, yeah, I would say we could find more species. I wouldn't be too surprised to find some more amidids.
But it wouldn't really like change any ratios or anything probably.
I mean, it would change ratios a little bit. I don't think it would change the discriminant function themselves.
Cool. Well, this is kind of just, I guess a little bit off the back of Matt's question there is, you know, I grew up in a place that has a lot of turtles, and one thing that's notably, yeah, the most turtles of North America. But one group of turtles is absent from gray, and I don't know very much about turtles, so maybe you can speak to this a little better. Where are the map turtles?
Oh, that's a good question. So Alabama does have an extremely high diversity of all turtles, but map turtles especially. Apparently, for some reason, though, map turtles are really tied to modic environments, like rivers and streams, which gray is not. The reason there's so many in Alabama is just because all those little river systems, well, you know, most river systems in the central continent flow into the Mississippi. So you have, what is it? The Graptomes, almost said Maptomes. Graptomes geographica and pseudogeographica are the main ones there. But then along the Gulf, you just get all these little tiny river systems, not, you know, tiny relative to the Mississippi, and they're each getting their own diversification of their own map turtle species. So map turtle, there's like proportionally 12 or 13 species of map turtles versus like 12 or 13 other species of diverkellids. So map turtles are extremely diverse because of they really like specific river basins and they don't like leaving them. The reason I don't think we have any at Gray is because Gray wasn't a river. I don't think it's habitat that's suitable for them. But we had a soft shell turtle, which surprised me. I remember the moment I learned there was a soft shell turtle here. We found one. It was Jared told me. It's like that was like my moment I'll remember for the rest of my life. So yeah, I mean, we could find a map turtle, but it's not something I'm putting money on.
Don't you think, though, the taphonomy of the site gives you an indication of the differences between the majority of the animals that we find versus an occasional trianid or map turtle? So, I mean, it's just as likely, don't you think, that we could find a map turtle, but not in the abundance that we find trachypes, not in the abundance that we find crispies.
Yeah, that was something you brought up in your comments actually, was the trianid may not have been actively living in Gray, or if it was, it may have, you know, wandered in there as an adult, kind of like we think the alligator maybe. But if there was like a stream or river system flowing into Gray, something like a Graptomes map turtle or a softshell turtle could be living in that environment and then just sort of got into Gray. I think really when we're talking about habitat preferences, an adult turtle, an adult softshell or map turtle or whatever, will just live wherever it wants. It's the babies that really are tied to specific habitats and environments. But once they get to a certain size, they'll just do whatever they want. That's the problem with turtles, they do whatever they want. It's hard to say, you give them rules.
Sorry to ask another question, but you just said something that made me think about conversations we've had about turtles. And is there any way to get at, could you run this treating adults and juveniles kind of separately? Because like you said with sliders, their diet changes so much as they age. And I think that's the case for many turtles. Could you treat juveniles kind of almost as a separate taxon from adults and see if that changes your results? Or do you think it would?
I'm not sure exactly how you do that, like mechanically, because not all turtles do that. So you would end up doubling up some of the species potentially. But there's probably a way to do it. I just haven't thought. Yeah, I did really want to. I mean, you could just add a second or separate ecological variable about ontogeny and whether or not they change as they age. That was another problem I had with classifying omnivores versus mesoherbivores versus mesocarnivores, is that pretty much all amidids eat nothing but insects when they're born, and then their diets change as they age. Chrysanes and Trachomenes move to a more omnivorous. They favor plants as large adults, but they'll still eat insects. Things like Pseudomes go from eating nothing but bugs to pretty much nothing but plants as adults. So I did use the adult diets mainly for mine. There's also things like Graptomes, like you brought up map turtles earlier. Female map turtles are well known, at least some of the species. The females eat nothing but clams or mollusks and mussels. Potentially because, you know, they're laying eggs, so they need extra calcium or whatever. But they have these huge heads used to crush the clams up. The males just don't have that. The males also live in different parts of the river, so they're not even using the same habitat. So you could do the same thing splitting, you know, males and females or adults and juveniles. That's the nuance of turtle lifestyles. Makes it really difficult to apply, you know, across the board rules to all of them in this way. So I tried to be as broad as possible, given as few boundaries as possible.
Do you have a question up there in the chat? Question for somebody?
Nope.
Oh, okay, good enough. I've got a question.
Well, I'm a
So.
loser. All right, well,
All right, so you mentioned some things like tortoises and landing turtle, Amidoidea, that today are limited to certain places. Do you think they might have lived in different places in the past? How might their habitat preferences be a consequence of modern conditions?
so I definitely think tortoises have always been restricted by temperature. I don't see any indication that tortoises were ever able to survive in cooler environments than today. And finding them at gray isn't surprising, given that it's so much warmer than to find tortoises that far north. The Amidoidea is a little bit weirder because you know, our modern interpretation would be the Amidoidea as it favors cooler environments. Today it lives around the lives around here. So this is where Amidoidea, mostly this 8.1 and 8.2 ecoregions. Defining it down here is a range extension that we wouldn't expect based on its modern ecology and environmental preferences. Now remember that between the time that gray existed and today, there was this thing called the Ice Age. And if you're paying attention, you would know turtles don't like ice. They don't like cold. They really don't like being cold. So during the Ice Age, a lot of these distributions would have been pushed. I haven't looked at the exact actual modeling. That would be another thing we could do with this, which would be interesting. But where those turtles ended up during the Pleistocene would be really interesting. And then as they bounced back, as the glaciers retreated, their ecologies, their distributions could be radically different from the way they were before. They could also have gone through bottlenecks. Matt, I think you had asked me a question about wood turtles.
Yes.
And what they did during the Pleistocene, and we found that there was a refugium of wood turtles down here in the southern Appalachians, where today there aren't any wood turtles. Now they live up here. So yeah, the distributions were very different during the Pleistocene and probably before the Pleistocene as well.
Well, so thinking about tortoises, not temperature, but in terms of, you had them all classified as open habitat.
Yes.
Are there tortoises today that live in closed habitats outside of the United States? And might that have been different in the past?
Yes and yes. So I think it's Indo-Testudo, which is more forested, but that lives in India. So the closest analogs that I had for proxies were gopher tortoises, desert tortoises, Gopherus. And I also looked a little bit at Testudo, although Hespero testudo, which is the other tortoise that we have classified at gray, it's probably related to Gopherus. It's in that Xerobatinae subfamily. But yeah, we don't really know what the tortoises were doing. My suspicion is they were probably living in more closed environments than the ones we have today, which is why I gave that caveat for the tortoises. So I think we have a better understanding of the aquatic turtles than the terrestrial turtles. Sean.
Bouncing off of that point, and I was going to ask earlier as well, so it ties in nicely. Did you look at specifically some of the South American and Central American tortoises?
The Californians. Not specifically. Remember, most of them live, yes, there are some in Brazil. Most of them live in Argentina and Chile, which are typically more open environments. But I didn't want to get too far out of the sort of taxonomic range of Gopherus. But I mean, yeah, that could be based on the morphology of the tortoises, which I just think we don't have enough of the fossils really to.
No, I didn't mean to infer a actual relationship, but more of an eco role. So like tortoises that do live in, more forested areas, maybe a little fossorial digging into embankments, eating fruit, things like that.
That could be. And I know that the Chelinoides do live in some sort of not wetland environments, but boggy, sort of not peatland, swampy environments where they walk really high up on their toes to get through the water. So depending on how high the precipitation was at Gray, they could be doing something similar. But that would be speculating. Which I don't want to do too much of it. I would love to travel back in time to watch that. My guess, though, based on the result that we got, there are tortoises down here. These are the Gopherus furlandii, Texas furlandii tortoise. So based on those comparisons, that would be the one that I would look at the most closely. However, if you look at, this is a picture from the Texas, Louisiana Gulf Coastal Plains, doesn't really look like Tennessee. It doesn't look like the Gray fossil site. It's not trees, a lot of trees, it's not elevation. So we're not really sure, we're apart from the aquatic environment, the terrestrial environment, how the tortoises were living there, is just a lot more speculative, I think. Hope that answered.
I think one of the things that we have to think about is the overall community here wasn't necessarily just a forested environment. And, around the pond, we have all these indications that that's correct, right? That it was really forested right around here. But we also have animals that would indicate that it may have been more open in areas, animals that would have engineered the environment into more open. What might be one of those animals that would help to engineer a more open areas within the forest?
I don't think the mastodon tunneling through the forest.
Yeah.
So it is possible that the tortoises were living sort of on that constructed
I think
environment.
we have to be really cautious when we just use our modern Holocene to make our analog out of, in that we have this tremendous lack of megafauna that are modifying the current environment that we base it on. And so we have to keep in mind some of those other big taxes, sort of like what you were bringing up too, is thinking about that broader environmental perspective of who else was in the landscape as well that may have altered it. So that's something to think about as a future direction, is looking at that broader aspect, because no proboscideans, that would be something else.
I can't fix that, but yeah, Gray's in the wrong spot. I don't know how that happened.
Resizing picture problem.
It's under the
Yeah.
spot, just not right in the middle.
It's actually right. It's right at that point.
It's
right there.
larger. It's larger than it used to be.
All right, it's still recording.
So Julian, kind of building off the idea of this Holocene baseline and what we're missing. So one of the things, you also said that, our understanding of aquatic turtle ecologies is much better than, our land turtle or terrestrial turtle ecologies. That said, there's a lot of things that have happened to aquatic ecosystems in the last, 200-300 years. They are massively changed. We probably know less about how those changes happen and what changes happen than we do about terrestrial ecosystems. And there's nothing we can do about it. Unless we. But how might we approach that from the side? Can you think of any good ways to get at a better baseline for these aquatic ecosystems?
Yeah, I can.
Go for it.
During my undergrad, my main project that I worked at and did with my undergrad advisor was zooarchaeology of unionid mussels, using archaeological sites where they've been collected and eaten and then disposed of in a midden. So we used that as a baseline for pre-industrialization Holocene unionids. And we did demonstrate, based on data that I collected, that there's a species of mussel that today only lives down here, but I was finding it in sites up here. So it's possible that applying that same kind of maybe zooarchaeological data collection to early Holocene turtles, also just turtle paleontology sites, fossil sites from the early Holocene, also historic records, possibly. One caveat I should make about the National Park Service database. So I only used turtles that were marked as present currently. And that doesn't necessarily reflect like bob turtles, for instance, could have been in there in the past, and they've just had so much range restriction in the last 100 or so years. So it is possible that there's some historic data that's missing there. So I was working with it.
Just identify turtles to something less, something more refined turtle.
Yeah. Yeah, I get a lot of, in my IDs of turtle specimens, a lot of them just say turtle shell. Good, thanks. Charlie.
So I think it's really cool that you did the community structure analysis with turtles when it has, mostly only been done with mammals. So has this made you think of any other non-mammalian groups that the community structure analysis could be applied to?
Yes, I think it'd be cool with other reptiles, maybe even all reptiles together as a community. Although there's so much diversity in ecologies there, it might not be feasible. Although there's diversity in the ecology of mammals, so maybe it is. Lizards would probably be good. Maybe amphibians, salamanders, or frogs. I think Blaine would know more about that. Unfortunately, insect fossils aren't, they just don't fossilize well. But I think that'd probably be the best possible. If we had a good, coverage of invertebrates, that would be the ideal situation. Invertebrates always have more data than vertebrates, always. We don't like that because we're vertebrate scientists.
Got specimens.
You ready? I probably.
Turn on your dot cam.
I don't know if that'll do it.
Blaine's been looking forward to this.
Yeah, I've been preparing all morning.
Yeah, you're excited. Is it the same that says happy birthday?
Oh, it's not for me.
Oh, happy birthday, Blaine.
Yeah. There's like, probably shouldn't blow.
Thesis defenses are just game shows.
And I've got number 4.5.
All right, we're gonna have to zoom way in on this.
Can you guys hear me at all?
You can see the grains of paper house.
Go with that one first.
So looking in front of the screen.
There you go.
Why could we flip it up so I can see the inside, the lingual side?
Oh, good, yes.
I believe that this is pleurodont,
Okay, pleurodont teeth.
which would make it not an iguana. Or
Okay.
sorry, not an iguana.
Not an
Or a
agave.
chameleon. Or a human.
Okay, what else could you say about it?
It's got very pointy recurved teeth. I'd say this is a carnivore, a predator.
Okay,
And based on specimens that I've seen, my immediate guess would be a varandid.
all right.
Or possibly a helodermatid.
How could you tell if it was a helodermatid or not?
I would think they would have some grooves on the teeth for indentation.
See any?
This is a high resolution. I don't see any currently.
Look up there again now.
Yeah.
If you look at the screen, you can see a little bit better now, I think.
So possibly, yeah. right here
Yes.
Could be.
Yes, that's what those are. Alright,
So my guess is, I mean,
so what is a helodermatid?
like a Gila monster or beaded lizard.
Okay.
It's an angry morph.
And where do those come from?
They come from, oh, like around here, and the Southwest and into Mexico. And I
Yeah.
do believe that we had a helodermatid at the Great Fossil Site.
That's right. So another thing that bird warmer. Didn't work.
I think we're getting the paper.
Yeah, I spoke on paper. You get the paper off the screen, maybe it'll get it for you. Oh, no. There you go, get that.
You can see it.
If you do stop screen sharing, I think hopefully people can see what's going on.
Okay. All right, let me know if you can see the specimens up there.
Want to go over and look at it on the screen?
That's probably better. I think it's better the other way.
Better the other way?
Some kind of cryptid.
Well, you gave it away as a reptile.
That's better.
All right, what kind of reptile is it? It's a reptile. What kind of teeth does it have?
Well, my first thought, it doesn't look pleurodont, but I know that sometimes acrodont dentition can have this sort of wall that comes up around it, so you can put pleurodont. It almost looks heterodont. These teeth look different from those teeth. It's very strange.
Yeah, I think it does. So it is heterodont.
And it's a reptile. It's not a turtle. I know that there are heterodont reptiles.
All right.
And
So-
let me look at the sides again. Very small. So can I ask,
Yeah,
is it pleurodont or is it the acrodont?
it's pleurodont.
It's pleurodont, okay.
So what, if it's from North America, what do you think it might be?
It's,
Pleurodont, North America.
it could be like Scaloporus or Tid,
Okay,
based on the size.
yeah, it's a tough one.
Yeah,
There aren't that many though that have a heterodont dentition like this.
yeah, that's what's really.
Crotophytus is one of them, the collared lizard.
Oh,
Yeah, remember that?
yeah,
They
that's an iguana.
Yeah, it's like one of it. All right, we'll go to number 2. I wanna. All right.
These are skulls, right?
I'm gonna zoom out here.
So on this psychopophyses.
See if we can do it so everybody can see it.
It's working.
I do not know verts very well, but I think this one is a snake.
Okay,
From the ones I've looked at.
why do you think it might be a viperiid? Do you remember?
I know they have this big process down here,
Okay.
but I looked at that many of them.
You did a really herb.
That was a while ago.
Yeah, so that's what that is. So you have a snake. Can you say if it's Procelus or
I'll say that this is Procelus.
what about the other vertebra? It's a very specialized one.
I believe that this is a.
No,
Two.
no.
All right, it's not. It's not that.
It's a strange one. Put it down there and let everybody see it. It's been a while since you've had paleo herp, I guess.
Yeah. The reptiles I know, their vertebrae have fused into a shell.
Not the cervical vertebrae.
Okay, so that's-
It's turtle.
Do you want a lifeline out in the audience? Anybody?
This isn't a turtle cervical.
Yeah,
Okay, I know that the turtle cervical vertebrae can go this way or this way. They can't go this way. So I think that's what is going on here.
so if that vertebra actually can tell you whether or not you're looking at a pleurodire or a cryptodire.
I don't know how to do that.
And so just like you were showing how they come
straight back into the shell, how the neck will come straight
Oh,
back
yeah.
in.
Yeah.
That strange saddle-like structure will allow us to do that.
Very cool.
Anybody remember that?
Yeah.
All right. Don't like vertebrae, I'm sorry. Teach all you out there to learn your vertebrae. Okay, that one is a lizard.
Salamanders are lizards. It is proceeds.
Yeah,
It's supposed to be a relatively not small lizard.
you remember what that bone is right there? Right, if you don't know.
Yes. This the top of it? Yeah.
That's all right, try next one. Try this one.
Any tiny little fragments here.
That was a quadrate, which is a really important lizard bone.
Osteoderms on here.
Yeah, so what he's looking at here is this one. Right there.
It's based on the presence,
And those are
the
osteoderms.
size of its osteoderms. I'm guessing that's another human anatomy.
Okay, good. So everybody needs to look for those when you're picking here at Gray. All right. Now, a little bit bigger bones.
The limb elements.
And what do they belong to?
If I'm not mistaken. Femurs, I believe.
I want you to tell what those two bones are.
I think this one's humerus. No, this one's humerus.
Yeah, you got it right.
And these are turtles.
They are turtles.
I believe they're tortoises. Possibly.
And Blaine, can you give him 4.5?
Yeah. See if you remember what he just said.
Yeah. This is a fossil.
So that one's actually a fossil. Put it under there so people can see it.
This is another femur.
They look awful similar, don't they?
They do, but there's differences right here back here.
Uh-huh.
So that's fine. It's probably the same system, which I think was the femur. Humerus,
Yes,
because that's,
humerus.
yeah, because the
They
humerus
they
is
can
larger
often
than
look
the torus.
backwards because the humerus will be larger than the femur oftentimes.
I do know that they're curved like this, so they can get around their shell.
Yeah.
They're bow-legged.
Humerus versus femur. Once you get them down, you can see, you probably want to spend a little time looking at those. And then finally, this last one here, let's show it.
This tortoise foot. Possibly. Has a foot.
Okay.
Is the front foot of the tortoise.
Okay, good. It's a tortoise foot.
Yeah, you know what kind?
Yeah, these were actually all the taxon that you mentioned.
First.
Yep. Next.
I got another one you can put up there while you're at it. It's a mammal.
What are you carrying around that crossbody?
Well, okay, how do I turn on this thing? Yeah, because apparently they're not seeing movies.
Oh. I don't know. You know how to do that, Chris?
No. I don't know that we've succeeded yet.
Keep trying to talk to you, Blaine, but you can't hear
His
me.
talk is still up.
Sorry
It's
about
not-
that.
It's not anymore.
Not though.
Might have to do it like as a conference call instead of a TV presentation.
Chris, just turn on your video and start the video graphic screen.
Yeah.
It's like that.
Well, that's true. I can try that.
Josh, I believe
They
this.
just see us, so maybe if we walk over here, jump up
I
and
can
down,
just,
Yeah,
come over here, get off your map. See what Wally says now.
so I think this is a rodent.
Yeah.
I can see that.
What makes you say that?
Well, it's got a large incisor, also has those, the sheep teeth. So it's the incisor.
It's working, you can see it now.
Could you tell me something about what kind of teeth those are? Anything that you could tell me about what kind of teeth they are, what this did?
Oh, they're high crown teeth.
Are they?
Oh, I can't see. I guess they're not. I don't believe they're ever.
Are they rooted?
Yes, not particularly well. I mean, you mean if they're in sock.
Right.
Yeah, you're in sock.
What is an ever-growing tooth versus a rooted tooth?
I know horses have ever-growing teeth. I believe. Those, they're still in a socket.
Still in a
They just
socket though.
Continually push up out of it. I guess, yeah, these are not ever-growing because they're. See, like, multiple sockets that they're in.
Nor
Obviously-
are horses actually. They're very high crown, but they're not actually ever growing. They have a root that's. there.
So there are some ever-growing roots, right?
So this is not one.
Anything else, guys? Oh, that looks good.
This is a gray.
Is it squirrel?
Yeah,
My squirrel.
this is a tree squirrel. It's a black squirrel.
Where's the squirrel?
Chris, do you have something?
Just one second.
Okay.
Read him out because I couldn't find the print. So you came to us liking, it's not gummy, but you came to us liking mussels. You did a thesis
Oh, no.
on turtles, so you're thinking kind of shelly creatures.
Uh-huh.
This is the wrong. There you go. So we've got some writing here, but don't worry about it, right? Here's a model. See if I can copy this. Just writing all over it. This is the wrong tab. Let's see if we can do. Open it up, something you can flip it around with. All right. This, oh, yeah, let's.
Here's the thing, over on the other, on this.
That thing you
Nobody
go,
will know.
say like PC or whatever. Yeah.
Now we'll work Julian.
Oh, that works. So yeah, there you go. The size is about this big.
This big.
Yeah.
I believe I know what this is.
You've got a good name on it.
Yeah, you know, finally. I didn't think we would.
Oh, it doesn't matter. Even though
We hear you,
Red
Wally.
said to have everything. I've tried talking to him, they can't hear us.
We hear
I don't know what the deal is. I don't know, because I feel like, what if this was a student trying to call in? I can use the chat. I can use it. I can remember to check it. Usually the first 10 minutes they don't.
Yeah,
And
because you're
so,
talking.
yeah, and so,
Typically not here on Fridays. I know Josh has a thesis defense going on downstairs. I
okay.
was gonna leave this box just in case Scott's not around, because I know sometimes that door's locked.
Oh, I see. Okay.
So
Yeah, click it on the icon.
just to make sure everything's okay with this.
We could have just left it on.
Yeah.
Gonna hear
I just-
Wally and Julie talking about things there.
Okay. I would say this is an osteoderm or scoop from a mammal. possibly, most likely South America, possibly North America.
Which is Kansas.
Kansas. Let's say this is from a xenarthran of the cingulate branch. Probably, if it's from Kansas, I would say that this is a glyptotherium.
Close.
It's a glyptodon, or is it the giant armadillo?
Yes.
I don't know the
Really
choice.
is. But it would be way, it's actually the northernmost record. for
Okay, that was
amphitheater. For
complicated that
anyway.
way. It is, and they're the turtles of animals. Yeah, I guess glyptodons are more evenly hexagonal.
Wait, what, was it a pompathier or?
Yep.
Okay.
I have one more question for you. That's just a question about the turtles. And if you were going to try and understand whether or not the gray fossil site box turtle was more like box turtle from Mexico, the aquatic version, what kind of analyses might you do?
Well, I would start with shell shape, maybe some geometric morphometrics. I would start by just comparing the Coahuilan box turtle to something like the ornate or Carolina box turtle, trying to see if you could establish clear differences with that. And then, which is basically what my first thesis was with tracking these. And then I would apply that to the box turtle here. Chris, could you use isotopes for that as well, to look at the diet?
You could, with generalist animals like that, a lot of times it's very difficult to tease apart. They usually don't end up in discrete groups, they end up kind of just a smear.
Okay. I don't know if there's any specific signals for aquatic versus terrestrial. I guess in that case, what you could do, maybe compare the tortoises to something like the Trachomenes. If you can see clear distinctions with that. And then if you looked at the box turtle, that might be an indicator of something like that. They're also, like, box turtles are not ancestrally terrestrial. They did evolve from, amidids, so their ancestors were aquatic. So it'd be interesting to try to identify the point in the past that box turtles did become terrestrial, like during the, in the process of the fossil record. And then comparing those ones to the great fossil site, morphologically again. I think there's a number of avenues you could take that are beyond the scope of this project.
All right, so I guess we're gonna ask everybody to step out. We'll talk to Julian for a few minutes. And then we'll send him out too.
Oh, yes.
I don't know.
What are you gonna do?
Well, you're not hearing anymore.
But actually, you turn.
Yeah, say thank you, everybody.
Bye.
Bye.